Today I'm going to be exploring the process, I guess, of me learning bioinformatics. So, bioinformatics to me is the process of some sort of model or statistics on things like genetics, diseases. The focus is usually drug development or some sort of product. Um, and basically, I created my same thing I always do, my little mill notes, and I process it in various different ways. These are old ones, so those don't actually go with this. I process in very different ways, and sometimes, unlike my other videos, which are very much cohesive and very organized, this is an example of a new concept. So the boards are not as organized in the beginning as it was. I think I started with this one, and then I had to then create other boards to then dissect different things. And they're both, all of them focus on different parts of something that has to eventually go into bioinformatics. So even for the information to go into the computer, it has to be um, transformed from the outside, which is the analog signal, into a digital signal. That's why I think of everything, including sound, color, etc. So there's different methods and methodology to do that. This is DNA extraction to database flow. So basically, this is where I start if I have any questions, and then other things sort of breaks it down. This one was focused on a concept of trying to solve a problem that is known to have some sort of genetic signature. And then finally, bioinformatics, I just, put a database in there and decided to go ahead and use synthetic data to try to understand how these things are processed. Now, I have another one that's not this one, but it's actually, uh, it's a larger data set. So with the larger data set, you can use machine learning methods on it. And basically, that's what the example was taking me through because of how much information that you get from each one of these samples. So this is a very tiny a sample size versus, a full on study where you will have a lot of data. And then that data gets condensed down and then you format it to go into the model. And I think it's called, what is it called? PCF or something? PCF, yeah. So PCF. This one is just a statistical. It's still, as you can see, log. So you're still dealing with computers. It's not like you're calculating this, and it's not definitely not calculated the way that a human would calculate this. This is very much computerized statistics, and it's comparing um, two genes, and it's doing the two-tailed test to see if the expression was significant or not. And I've never seen a volcano plot before, but this is a volcano plot. And the middle is there's no significance, and then the other outside one are outside of that. So those are moving towards a particular value. And here's the significantly, specifically the plot shows the gene expression change measured by absolute value of log 2. Fold change increases the statistical significance also tends to increase also. And I have some notes about what the volcano plot is, what it means, all that good stuff, etc., etc. For this, I think this is very simplified, very, very simple. The data that I saw on a lot of these gene databases and stuff, it's hard to read, and you have to get used to the software, and you also have to get used to um, sort of the labels and stuff. This is the most comprehensive I've seen, like pretty organized. These data sets, and you're able to do a lot of analysis in the portal itself. So, yeah, but there is also just a lot of information that you have to understand about the data set. So you have to really pay attention to the labels and what they mean. And it's very much more technical than a lot of the other stuff. When you're dealing, for me, when I'm dealing with stocks, quantum things, business, the labels mean something very specific. And genetics, genes, yeah, it does too, but you have to sort of understand the sequences and how they have put the genome together, their position and the names and everything else. And you have to make sure to keep everything very much cohesive and not confused, especially when you're doing multiple data sets from various different experiments or results. So that's pretty much it for that. This ties into my exploration of some of the new biomedicine techniques and how that's going to impact. Also, it ties into my anthropology, which also deals a lot with DNA because unfortunately, it's a lot of computational work versus actual research when it comes to people. I do think it can help render a result, but unfortunately, the way I'm seeing it, the framework, is a lot of people are pushing just for it to be informatics, bioinformatics. It's already bioinformatics. And they primarily just focus on what they can put into the computer. And these are like more like pointers or tools that can maybe point you in the right direction, but it cannot solve bigger problems that are much more complex. You are also dealing with a technology that is very much restricted in how we computate things. So, at best, it is a tool that should be used along with the traditional anthropological tools. Also, with medicine, I think making sure that we're very much close to a workflow with our environment and not being separated too much from the environment. You have to start thinking about how medicines were created and how these happenstance most of the time did not happen always in a restricted laboratory. These things happen through our environment. These things appeared and then people created synthetic versions of these types of medicines, herbs, things like that. A balance is what's needed. So, laboratory and a balance between those two together. And people should always be connected to their own health and their own healing and understanding, and not just a small group of people somewhere who are so far from the actual environment that it just doesn't make sense to even spend the time because you're going to get the same results over and over again. That's pretty much all I have to say, and I'm hoping that I explore more and I get to see you guys' projects too. Until then, bye.